Welcome to my channel Adhyan with Flow. In our previous video we discussed about interphase. Now let's start from M phase. M phase is dividing phase. Now preparation is complete. Now cell want to divide. First karyokinesis. Firstly nucleus will divide and after then cytokinesis cytoplasm will divide. So this M phase divided into four phases: prophase, metaphase, anaphase, telophase. Now what happened with prophase? Let's start. What is prophase? Starting phase of division. Very starting phase of division. All kinds of protein are complete. All types of cell division is in cells enzyme is complete. Now it's time to dividing. Now what happened in prophase? Chromosomal material condenses to form compact mitotic chromosome. Like in starting phase, chromatin. This is like a bead in structure, thread like structure. Starting off prophase, they compact. Chromosomes are seen to be composed of two chromatids attached together at a centromere. Now this compactness is look like this, and this is the centromere, and this is the chromatid. These are the chromatin. Okay, now centrosome which had undergo duplication during interphase, centriole already duplicate. Look at me, centriole already duplicate in starting of S phase, interphase step, begins to move towards the opposite pole of the cell. Now this centriole goes to different pole and this centriole goes to different pole, like they arrange like in this manner. This is the cell, there is one centriole and here is another centriole. They reach at different poles. Now they arise some microtubules, esters. They attach with, look at me, they attach with centromere of chromosome. They attach with centromere of chromosome. Look at this, look at this, look at this, look at this. Each centrosome radiate out microtubule called esters. The two sisters together, the spindle fibers form mitotic apparatus. Look at here, guys. In early prophase, this is the phenomena. First step is nuclear envelope start disappearing. Already is written here, guys. Already all thing is written here. Firstly, the nuclear envelope is start disappearing. This is the cell now, guys. This is the cell. This is the nucleus. Of course, nucleus covered with their nuclear membrane. So this nuclear membrane start disappearing. Alright. Now these chromatin are thread-like in structure. These chromatin are thread-like in structure. Now they compact. Look at it. Now they compact. Now they compact and make a chromosome. Look at me, guys. This is in this structure, thread-like. Then they condense. Then they more condense. This is the centromere. This is the centromere. Look at me, guys. This is the centromere. Now understand. Now betterly understand. So this is the centromere. Centromere. This is also a chromosome. This is the centromere. And this is the chromatids. Okay. This is the sister. Now look at their nuclear envelope. Look at their nuclear envelope. They start disappearing. They start disappearing. Now chromosome condensed. Cells end of the prophase when viewed under the microscope do not show Golgi complex. There are negligible Golgi complex. Golgi complex is removed. Endoplasmic reticulum, this is disappear. Nucleolus and nuclear envelope, these all things start disappearing. Start disappearing. I repeat again. Starting off prophase, what happened with them? When prophase start? After G2 phase. After G2 phase, cell enter in M phase, and M phase divided into four parts. They enter in prophase. And what happened with process? Their chromatin, thread-like structure, they compact in this manner. They also compact in this manner. They further compact like this manner. So this is the center point, centromere, and these are chromatids. This centromere attached with some esters. Esters connected by centriole. They bind with centriole, because ester is connected with, this part is connected with centriole and this part connected with centromere. Here is another centriole, this part is connected with centriole and this part connected with centromere. And next thing is nuclear envelope disappear, nucleolus start disappear, endoplasmic reticulum start disappear, and Golgi complex start disappear. So this is all phenomena occur in prophase. Now let's move towards the metaphase. What happen in metaphase? All chromosome, look at there, they arrange like in this manner. Look at guys, they are microfilament arranged like in this manner. They attach with, look at here, they attach with their centromere. All microfilament attach with their centromere, so they can pull. See, they can pull. And here, look at here, this is the centriole. Look at here, they duplicate their self after S phase. They duplicate their self. Now, in this phase, what happened? These all chromosomes arrange in a metaphasic plate. Look at here, guys, they arrange in metaphasic plate. Look at here. Till now, all kind of cell organelles disappear, all kind of nuclear envelope disappear. Look at here. So these are spindle fiber. Look at here, guys, I'm again repeating, these are centrioles. Okay, these are spindle fiber, they connected with the centromere, the center part of the chromosome. Look at here, guys, all spindle fiber connected with chromosome. Here same thing is here. Here also centriole present, and these also connect with the centromere of the chromosome. Look at me, guys, here. This is the chromosome. This is the chromosome, guys. Here, this is the spindle fiber, and this is the spindle fiber. This spindle fiber connected with another centriole. This spindle fiber connected with another centriole. This is the arrangement. They connected with the centromere, center part of the chromosome. This is arrangement. Here, this is the arrangement, and this is the metaphasic plate. They arrange in center like this plate manner. We call it as a metaphasic plate. We can also say metaphase plate. The plane of alignment of chromosome at metaphase is referred to as metaphasic plate. Clear, guys? All clear? Look at here. A small disc-shaped structure at the surface of the centrosome are called kinetochore. Here, at the centromere, a disc-like structure is present. We call it as a kinetochore. These structures serve as a site of attachment of spindle fiber, and here spindle fiber will attach. We know now this is the centromere, center part of the chromosome, and this surrounded by disc-like structure. We call it as a kinetochore, and these kinetochore attach with spindle fiber, and spindle fiber attach with centrioles. Okay, formed by the spindle fiber to the chromosome that are moved into position of the center of the cell. The metaphase is characterized by all the chromosome coming in line to equator. Yeah, they arrange in equator. Yes, guys, with one chromatid of each chromosome connected by its kinetochore. Yes, guys, they connected with their kinetochore to spindle fiber from the one pole. Look at here, this is the one pole and this is the another pole. One pole and opposite side of pole they connected with. Okay, spindle fibers attached to kinetochore of chromosome. This is the special feature of metaphase. Chromosome are moved to spindle equator and get aligned along metaphase plate through spindle fibers on both poles. Look at here, guys. This pole centriole, this pole centriole, they connect. They give spindle fiber and they connect with centromere, kinetochore. Okay, this is the metaphase. Now anaphase, what they do? Now they centriole, what they do? Now they push our spindle fiber towards itself. These spindle fiber, they push their, these centrioles also push their spindle fiber towards them. So now at the end of the anaphase, this kind of structure I'm drawing for you. This is the centriole, this is the spindle fiber. This is the centriole, this is the spindle fiber. Now what they do? They push chromosome. They are strongly bind, huh guys? They push send. Look at me. Here I am drawing something for you, and now you can betterly visualize. They push spindle fibers towards them. Here force and here force. Force is created by centriole and spindle fiber moves towards the poles. Now this arrangement of chromosome now break. This arrangement of chromosome will break, and now finally they separate. Now finally they separate and they goes towards the different pole. Born na guys, they are connected with their connected, so not easy for separate them, but spindle fiber push towards the pole and they separate. So centromeres split and chromatids separate. Chromatid moves to the opposite pole. Look at here, they move to the opposite pole. This is the feature of anaphase. At the onset of anaphase, each chromosome arranged at the metaphasic plate, it split simultaneously, and the two daughter chromatids, now referred to as daughter chromosome of the future daughter nuclei, begin their migration towards the two opposite poles. Now these chromatids moves towards their opposite poles. This is the feature of anaphase. Now let's talk about telophase. Now telophase feature is chromosome cluster at opposite spindle fiber and their identity is lost as discrete element. Now they move towards the pole, and now these chromosomes, they reduce their compactness and they make chromatins. They lose their compactness. Now chromosome lose their identity and they appear like this. They simplify their structure. Nuclear envelope develop around the chromosome. Look at here. Here nuclear envelope start appearing. Nuclear envelope start appearing. Cluster at pole forming two daughter nuclei. Now two daughter nuclei also form because they segregate now. Look at here, two nuclei, one nuclei, and this is the another nuclei. Now future they look like this. Nuclei is separate, so within a one cell, two nuclei will form. This is one nuclei and this is the another nuclei because they have their chromosome. Now nucleolus, Golgi complex and ER reform. Then the Golgi complex ER will reform. Now cytokinesis will occur because nucleus is formed, their organelle is formed. Now their cytoplasm, they start dividing. Now they divided into two cells. So we started with one cell and one cell divided into two cells. This cell phase, prophase, metaphase, anaphase, telophase. At the end of telophase, their nucleus look like this. And now finally this. Okay, this. Now this divide and make a two cell. This is the one cell, this is the another cell. This is the one nucleus and this is the another nucleus. This is their centriole and this is their centriole. Again, if they want to go into prophase, then this centriole, what they do? One at this pole and another with this pole, and they start duplicate. And they start duplicate their self. And now they show metaphase, anaphase, and they again split. So cytokinesis means mitosis accomplish not only the segregation of duplication of chromosome into daughter nuclei, but the cell itself is divided into two daughter cells. Now they make two daughter cells because in starting here is nucleus is divided, not complete cell is divided. But now cytokinesis, complete cell divided into two parts. Now this separation we call it as a cytokinesis because a cell plate is formed. Okay, division get completed. In an animal cell, this is achieved by appearance of a furrow. They make furrow in the plasma membrane. They make a furrow. They make a hole. They make a like this. This is the notch. They make a furrow in the plasma membrane. The furrow gradually deeper. They move deeper. This is the furrow, guys. This is the furrow. And now this furrow deeper and deeper. more deep, more deep, now more deep, and then divided into two cells. Okay, enclosed by relatively inextensible cell wall. There will go undergo cytokinesis by different mechanisms. In plant cell, wall formation start and the center cell grow outward of the meat existing. They form cell plate. They form cell plate. Plant cell make cell plate because they have hardest cell wall. Animal cell does not contain any kind of cell wall, but plant cell contain cell wall. Farrow is easy in animal cell because they have PM, plasma membrane, but farrow is not possible in plant cell because they have cell wall, guys. So they need plates. They want rigid structure. So in plants, cell plate is formed, but in the case of animal cell, a furrow, a notch like a structure will form that represent the middle lamella between the walls of the two adjacent cells. Okay, this is the middle lamella between the two adjacent cells. At the time of cytoplasmic division, organelle like mitochondria and plastid get distributed between the two daughter cells. Yeah, of course, all cell organelles distributed in their equal amount. In some organisms, karyokinesis is not followed by cytokinesis, as a result in which multinucleate. If cell is still one, but nucleus get divided. This phenomena we can call it as a multinucleated phenomenon, and this division is called it as a syncytium. This division is found in Drosophila. In the case of plant, this division found in endosperm of coconut. So now this is all about M-phase. Now there is some significance of mitosis. Mitosis, or okay, we will discuss in our next video. Mitosis importance and meiosis starting we will discuss in our next video. So thank you guys. Thank you for listening me. Keep smiling and revise. And subscribe guys, subscribe my channel. Thank you.